the isolation of antibiotic-producing bacteria from soil with an in situ diffusion chamber.
Nicole, are you what?
Dr. Kaiser. So I'm working kind of in Michael's kind of similar lab. To start, antibiotics allow for the treatment of infectious diseases and advancement in invasive surgeries. Misuse and overuse has led to the development of antibiotic-resistant bacteria. These bacteria acquire these through antibiotic-resistant genes via horizontal gene transfer and natural selection. About 700,000 people die per year, and it's estimated by 10 million per year by 2050 die of antibiotic-resistant infections. Antibiotics are sourced from the environment, such as soil, which is what we're going to be looking at. The problem is only 1% of microbes will grow in standard laboratory conditions. And previous studies have done modifying the media, incubation conditions, structural designs, and to achieve more bacterial diversity. So the goal of all of these is to mimic the natural environment of the microorganisms as closely as possible in hopes of growing novel bacteria within the unknown 99%. This is our approach. It's a diffusion chamber, so it starts with a stainless steel washer, and there's nutrient agar in the middle, and we take a soil dilution and put that onto the agar, and then we sandwich it between two polycarbonate membranes, and these are held on with silicone glue. This is what it looks like when it's put all together, and then this is what it looks like when you put it on top of soil in a petri dish. And the idea is that there's growth factors from the soil that will come up through to the agar, and the soil dilution that's on the agar is the same, it's taken from the soil that the diffusion chamber is on. Why use polycarbonate plastic? It's transparent, so we can see what's growing within the chamber. It's a thermoplastic, so it can be autoclaved multiple times. It's pliable and durable, so it can be taken on and off really easily. It's hydrophilic, so it attracts water, so it helps things from the soil come up through the membrane easier. And then this is the standard for previous studies. I just wanted to note that Bowman, Lewis, and Epstein were the first to do the structural design, so ours is based off of theirs. Up close, the polycarbonate is manufactured to have pores that are 0.2 micrometers across. This is referred to as general sterile filtration because it keeps out most bacteria when you filter things through it. What does pass through is water, ions, metabolites, chemical signals, and growth factors from bacteria within the soil. This delivers the possible benefits of co-culturing without overgrowth of the inoculated area. So clearly we've come very far. This was October of last year. This was our most recent experiment in March. Very big progression. The initial obstacles that we had to overcome was we had very small washers. We couldn't find the appropriate size, so it was hard to see what was within the chamber. We didn't have the right membranes for a very long time. They kept telling us they would come, and then they were on back order, that they would come, they were on back order. So we were using these paper membranes that were just already in the lab. They fall apart easily. You can't see through them. This prevents us from putting the top membrane on most of our initial diffusion chamber experiments, so that's another limitation. The soil would dehydrate very easily because you're incubating it at 35 degrees Celsius. This in turn deflated our agar, so our agar got very dried out and all the water went out of it, and so nothing grew on it. To solve the problems of dehydration, we initially put the diffusion chambers within this plastic container that had wet paper towels in the bottom, and then you take the whole container and put it within the incubator. And the idea was it was circulating humid air to keep everything hydrated. And this worked really well. So our soil, after a few days, is clearly still wet. There's agar with little small colonies growing on it. We had bigger washers now, so we can see what's going on. So this was our 1:100 soil dilution. The only issue with this was our control looked almost identical, which is not good for a control. This should have been sterile. It shouldn't have anything growing on it. And we tried a lot of different methods within the lab to try and prevent this contamination, but it kept happening through multiple experiments. Obviously, not being able to put that top membrane on is probably the biggest issue that contributed to the contamination. So we have our diffusion chambers in that plastic container. There's humid air coming in contact with soil, coming in contact with the agar surface, so microbes are most likely landing on it and growing. Simultaneously, we realized that having that agar within the diffusion chamber and then putting the soil dilution right on top wasn't sustainable in the long run, because if you have a humid environment, the agar's wet, so the bacteria can spread really easily across the surface, which isn't ideal if you're trying to isolate specific colonies. And then we knew eventually that when we got the correct membranes, if you put the top membrane on and then you try and peel it off and all of your dilution is on top of the agar, it's just gonna peel off all those colonies with it. So we began mixing the dilution with liquid agar, and then so once we poured it into the diffusion chamber, it would get solid and then the colonies were embedded within. We finally got the correct membranes, we improved our aseptic technique even further, and then we started parafilming the plates instead of having them in that container to reduce the circulation of the humid air. This was our result. So this is a 1:100 soil dilution mixed with nutrient agar. This is the same solution we poured into the diffusion chamber, but we also would pour it into a petri dish as well and incubate them alongside each other, just so we can see like a clearer image of what's going on. So there's colonies on top, colonies on the bottom. There's all these small colonies embedded within, which is the most important part, because we're assuming that these colonies normally would not get a chance to grow if you just plated everything on top of the agar, because all this fast growing bacteria is competing for the surface. So that was a really good thing to see. And then I just wanted to bring this diagram back because all the next pictures are this small section of the diffusion chamber, because we're actually able to pop it out and look at it a lot better. So this was our 1:100 dilution. We had those same colonies embedded within the agar, even though very small. We also had some bigger growth right there, but we're not really sure what that was. But. And then I also wanted to note that as another control, we took that same mix of 1:100 dilution with agar, put it in a diffusion chamber, and then put all of that on top of nutrient agar instead of soil. We still had all those colonies embedded within the agar, which was good to see, but also what this is most important showing is that the agar stays white. Besides, when we put it on top of soil, it turned yellow. So this is showing that those metabolites, ions that I talked about, there's something coming up through the soil, changing the color of the agar, so hopefully the diffusion chamber is doing what we want it to do. And what's important about this is that our control, all those small dots are just air bubbles when we zoomed in really close. So we had no growth in our diffusion chamber, that was our control. But the color still changed, so it's showing that anything coming up through the soil isn't contaminating what's in the diffusion chamber. In summary, we saw problems with dehydration, contamination, and visibility. Colonies were successfully suspended in the agar without overgrowth of fast-growing bacteria. And changing color of the agar when incubated on top of soil demonstrates diffusion of small molecules from the soil through the membrane to the inoculum. For future work, we want to test these colonies picked from the diffusion chambers for antibiotic activity against antibiotic-resistant pathogens, which is the whole basis of this experiment, but we just haven't gotten to that point yet. Our next steps is we're going to do decrease the dilution, so hopefully we get more of those colonies instead of just a couple that we saw. And then that's my first sentence. Yes?
Good talk, Emily. If the intent is to figure out a mechanism for growing unculturables, right?
Mm-hmm.
How do you know that the cells that come up are unculturable? What's your control to figure out whether the ones that are coming up wouldn't have come up?
Mm-hmm.
Do you have a control that's somehow, azenic, not exposed to soil, or otherwise? You see what I'm saying?
Mm-hmm.
Yeah. I mean, I can see it's that level of control. I just wonder if you have thought about how to discriminate against those.
Probably like this picture I had earlier. The one where we put it just on top of agar, so nothing's changing besides having soil underneath versus no soil. If we get different growth between those two, it's hard to determine like what is different growth, because greater diversity, greater amount of bacteria doesn't always mean we're cultivating new bacteria. That's a really good point. I would say this would be a control, and then also like the floor plate would also be a control. and just comparing, like, because we're not sure if we're gonna do, like, we could do sequencing and see exactly what's in there because they're from the same soil dilution, or we could test it against different bacteria. It's kind of like hit or miss. We're kind of just like picking random colonies and seeing what works. So it's hard to definitively say what's in each, like what exactly comes out of it, if that makes any sense. Yes.
So you mentioned that the colonies that you were most interested in were the ones that were embedded in the matrix rather
Mhm.
than on the surfaces. Is it? I will fully disclose that I am not a biologist. Is it possible or feasible to like selectively get that like stratum of the internal part of the agar without getting anything that's on the surfaces? Like, can you selectively get the organisms that are embedded in the center?
I'm not sure. I think with the whole point of like the ones in the center are the most important is because once we do peel off those membranes, we are peeling off that bacteria that's on each side. So that's why we're most interested in the ones in the middle. And there's also the factor of how much oxygen they're getting, because if they're embedded in the agar, I know there's diffusion of oxygen molecules throughout, but they would have less oxygen than bacteria on the surface. But I'm not sure how to like selectively get those besides, I don't know.
Like trying to scrape selectively, or
Yeah, eventually we're gonna have to pick through the agar and like cut it open, but we're just not at that point yet,
Got
so. Yes.
So it sounds like you said that your methods were based on an article, I think it was from like 2007. Are you planning to publish your methods? Because it sounds like you identified some problems that, or did like that article from 2007 have the same, did those researchers have the same problem? Because it sounds like you found some big problems and found some solutions, and it seems like that might be useful for the field. Again, I say this as somebody who has zero knowledge of any of this.
Yeah, when you really do research and you're like, I'm gonna do this experiment and you decide what you're gonna do, when you're actually physically doing it and figuring out all the kinks, you realize that they don't put a lot of that in the article. So it was hard to figure out where they got things from, how they even autoclaved it, because I'm using glue, so I can't like autoclave my glue and then use it because it'll probably mess up how like sticky it is. So it's a lot of trial and error, and that's the thing that's been probably most frustrating is kind of like teasing out, because I'm sure they ran into some of the same issues. I saw some articles mentioning dehydration, didn't talk about how they solved it. The plastic container was Dr. Kaiser's idea. I know some of them parafilmed, so that was why we started doing parafilm. But yeah, it's a lot of like guessing, trying things, seeing what works. So that's why we're more like proving a concept, because I have a DIS student I'm working closely with, and once we figure out all of the issues and we're seeing growth, then she's gonna try different types of media, because we've been stuck doing nutrient agar because it's the most basic, but trying different media, trying different conditions, like all that other stuff. So it's more of a proof of concept, and I thought we would kind of get more to the antibiotic discovery so far, but research kind of tends to stretch out a lot.
So do you plan to publish the proof of concept though? Because it seems like if you went through all of these steps, if everybody who's trying to do this has to go through all these steps and figure it out on their own, like wouldn't it be useful to say, no, we figured this all out, here's how you can avoid these, go straight to the chase, so other people can move it forward, you and other people.
Yeah, I agree. I think that would be very helpful. For sure. And the membrane thing was another thing that was very limiting, because we ordered them in November and they said they were going to come in December, and we were like, perfect. And that was when I was like, everything's kind of ramping up. They were on back order to January, then to February, then to March, and we ended up just getting a different pore size than what I wanted, because I wanted a smaller pore size. So it was kind of like, we're running out of time, we kind of got to get this going. So, but it was nice because we figured out a lot of the dehydration issues with the paper membranes, which are cheap, because the ones that I got are like, I had to apply for a CSRF grant for those, so, yeah. Yes?
Great job, Emily. Do you plan on continuing your research, like going to a master's program and testing some of your cultures?
To be honest, I don't think so. I know Olivia is continuing this project for her honors thesis, so it'll be interesting to see what she finds, because it'll be a whole nother year of her doing trial and error stuff with it also. So.
Great.
Awesome. Thank you.